So today, I'm very pleased to introduce Kris Raghavan, who is a PhD candidate at Drexel University, and he works and has been working for a few years with my friend and colleague Edna Cukierman at Fox Chase University. And he's going to talk about Netrin and functional characteristic of stromal-derived extracellular vesicles in pancreatic cancer.
Hi, everyone. Thank you so much for having me here. Good morning to some, good evening to others. I'm really excited to be here today presenting my PhD, what I've done so far, working towards my PhD. I work in, Dr. Vizio said, in the laboratory of Dr. Edna Cukierman at Fox Chase Cancer Center in Philadelphia, where the ICEV meeting was supposed to be this year, but now will be virtual in about a month, or a few weeks actually. So this was going to be the title of the poster that I was going to present there. I think it still will be, but I'm really happy to share with you my talk, Casting a Small Netrin G1, the Functional Characteristics of Stroma-Derived Extracellular Vesicles in Pancreatic Cancer. So I want to firstly start off by going over the cellular context in which I'm doing my EV investigation. I'm going to be touching on the challenges that exist understanding pancreatic cancer, as well as introducing the perspective that our lab takes studying this disease from the tumor microenvironment, specifically from EVs being secreted from cancer-associated fibroblasts. And then, get into why I think this study, using EVs can really shed a lot of context onto, shed a lot of light onto, understanding the disease progression that exists with pancreatic cancer. Then I'll be getting into the focus of my talk, which is what I will be looking at, using the function of cancer-associated extracellular vesicles in PDAC, specifically looking at the functional role of Netrin G1, the protein that our lab is really excited about. And then finally, I'll be getting into what I think is potentially the most exciting in terms of how I would like to contribute to the field of EV science, identifying distinct subpopulations of EVs. I also want to add, this is a really hugely educational moment for me. I don't often get the chance to present my work to people with a large group with an EV background. So, also towards the end, I would like to pose, some questions and some thoughts that I've been having, really like mulling over, my investigation, comparing it to what I've been reading and writing about and watching these talks. So, thank you again for having me. Oops. Okay, there we go. So I just want to come back briefly to this image. It shows the human pancreas during PDAC. This image was made using immunostaining and then using computer-generated masks to highlight unique cellular components within this tissue slice. So this image was generated by a really fantastic microscopist in our lab, Nalima Shah, who sadly passed away last summer. I like to show this image not only because it's really beautiful, but I think it really brilliantly highlights the overwhelming complexity that exists in the tumor environment and just like, in organs in general in a disease state. There is a lot going on here. And I think it really, brilliantly illustrates just how much we need to understand about this disease that is so terrible. And it is very terrible. It has a very poor survival rate. I'm sure most people are aware that the 5-year survival rate is very, very low compared to most other cancers at about only 9%. It is relatively rare as a cancer. However, it accounts for 3%, or the number 3 cause of cancer deaths, falling behind lung and colorectal, which are much more prevalent, as many people know. And the reason for this, I think, is most people don't even know that they have this disease until after it has metastasized and spread, as you can see here by percentage of cases, by stage when they're diagnosed. So, if you look at that breakdown by how these 5-year survival rates relate to when this disease is found, you get almost a 10-fold drop from the localized to when it has spread throughout the body. And of course, it becomes really impossible to treat at that point. So thus, there's a clear need for better ways to detect PDAC in humans and potentially treat it at early or pre-metastatic stages, which is what I hope my investigation into this disease from looking at using extracellular vesicles as a tool will really hopefully elucidate. So I started my PhD project in about 2016. There's been tons and tons of EV studies that have come out since then. Obviously, PDAC is just one cancer, but there's been tons of other cancers. Not only that, but in almost every physiological context, there are studies revolving EVs. But I think a lot of them kind of hinge upon looking for biomarkers, understanding the cargo, metabolites, RNAs, proangiogenic factors, EVs being able to affect chemoresistance in cancer cells. But I think a lot of studies, or the majority of studies that I've come across, at least in the last few years, have been really centered around understanding the tumor, the cancer cell itself, as opposed to studies that, there are a lot less studies that I noticed that are surrounding the tumor microenvironment, like, the cancer-associated fibroblasts are like this in the bottom right, like the natural killer cells. And we know that all, cells produce extracellular vesicles. And when we think about the system that these organs are being, interacting together and a part of, I think it becomes really important to think about, the specific disease and how, this microenvironment and, all these cells are interacting together. Specifically in PDAC, I want to, take some time to show why our lab takes the microenvironment, takes the perspective from the microenvironment, and why, it's so significant in terms of our study. So here you're just going to see a simple illustration of pancreatic ductal epithelium and the microenvironment or stroma that supports it down below. So these factors, there are a bunch of different cells that include, macrophages, immune cells, nerves, fibroblasts, and, specifically the extracellular matrix that these fibroblasts are producing to support and cradle this system. What we see as very characteristic in early disease progression are these neoplastic events, or what we call pan-ins, that are accompanied by a dramatic activation and restructuring of this stroma. And eventually this progresses to full bona fide carcinogenesis, wherein the tumor microenvironment has changed so significantly and is now very tumor supportive, can also be immune suppressive and promote inflammation, as many studies have gone into. But what I really want to highlight here is that these changes in the microenvironment are happening before you even see like the tumor starting to form, which is why we really believe that if we can understand, the cellular context, that's, the changes that are happening at this stage. And, for me, thinking about, looking at EVs as snapshots of, cellular contact, a cell at any given time, we really want to be able to understand, these changes that are happening at this point. Now, I think to appreciate those differences, I want to show you a section of what the actual, pancreas looks like during this change. So you see here, we have a tissue section of a non-cancerous area of the pancreas. Identified in orange is the epithelium, and in white is the stromal content, which is composed mainly in this state of as quiescent fibroblasts and the secreted extracellular matrix that they're producing. But during the transition to PDAC, as you see here, this really stark contrast, you have this really dramatic change occur in the microenvironment. These fibroblasts are becoming really highly activated and start producing huge amounts of extracellular matrix, which when we look at a resected tumor mass, up to 80 to almost 90% of that tumor mass can be comprised of just stromal content. So I think that this, when you think about it that way, it really establishes the significant influence that these cancer-associated fibroblasts have on the progression and support of this disease. Also, these CAFs, we understand, in this disease state to be very phenotypically distinct from that of the normal inactive fibroblasts, and this has implications, of course, for the different types of EVs that they are secreting. And as I kind of mentioned, that the extracellular matrix itself is structurally very different. So if we were to zoom in on these white regions, we would see something pretty interesting, I think, which I'll show you through the use of fibronectin staining. So fibronectin, this is immunofluorescent labeling of fibronectin, which is a primary component of the extracellular matrix. You're seeing that in white. And we see that inactive fibroblasts produce a very loose, unorganized matrix, kind of looks like very spiderwebby. And that looks very different from what you see in the architecture of a matrix generated by cancer-associated fibroblasts, whose fibers are very stiff and very highly aligned. Now, our lab uses these differences as one means of validating the activation state of our fibroblasts in vitro. And we do that by quantifying the angles that they're aligned by using a program in ImageJ to measure these fibers and what angles they are oriented at. And then we can put them all together to see that our cancer-associated fibroblasts are producing a very highly aligned matrix, and we're able to quantify that pretty effectively. The reason why I'm adding this now is because I find this to be a really particularly important aspect of the biology here in the context of extracellular vesicle research, because it means that we must not only think about the biogenesis of EVs and their cellular uptake, but there are a lot of other extracellular factors that can influence the fate of certain EVs, the matrix just being one of those. But of course, it is so prominent in this disease that it is a very important thing to think about. So another important point, I think, especially in the context of extracellular research, is that when we talk about EVs having the power to act as snapshots of the cell that they were secreted from, it's really paramount that we culture these cells to be as physiologically representative as possible to how they would behave in the body. So there are a lot of different, methods of doing this. For epithelial cells, some people use organoids. There's the technology of organ on a chip. But for studying cancer-associated fibroblasts like we do, cells that are building this three-dimensional extracellular matrix, we need to culture these cells in a way that they're able to do just that. And so we start with patient-derived CAFs that we plate and culture over the course of a week. So we will plate them at a very high confluence in the beginning so that they don't waste any energy spreading out, and they can just start growing up and producing matrix. And so we give them ascorbic acid treatments daily so that they strengthen this matrix and all the architecture there, so they can build, keep building and building and behaving really healthy. We understand that as EV researchers, we have to switch to condition, like an EV-free media to condition for 48 hours. So I'll do that around day 5. And then at the end, I'll have my conditioned media that I'll be able to harvest EVs from. But just so that we all kind of, this is just one, a simple schematic, but to really appreciate, I think, the difference between day 0 and day 7, this other schematic here will show that, we have this monolayer here we start with, and by the end, we have this, really like multi-layered architecture and that these CAFs are existing in. And so you think about these CAFs that are up here, they're very far away from the plastic, and they have this nice environment that they're sitting in, and they're going to be much more physiologically representative of what we would see in a body. And as one small hint to or small example of why that's important, I'm just going to show now, before I get into the real significance of Netrin G1, that when we culture these cells and we see their expression profile in 2D versus in 3D, we see this Netrin G1 protein significantly upregulated, really highlighting that this in vivo like 3D culturing system, is really important for letting these cells behave as they would in the body. And I think that applies to pretty much every different type of cell in vitro work. You always want to make sure that you are doing your best to represent in vivo like circumstances as best as possible. So with that, like I said, we have our conditioned media. I use differential ultracentrifugation, like many, to enrich my final pellet in exosomes and similarly sized EVs. So to do that, of course, we must validate, based on morphology and size, as well as through biochemical markers and through our NTA, nanoparticle tracking, just to make sure that we are enriching for everything that we want. And I feel, pretty good about that. So now that we have our raw material, we can start asking some questions. Specifically, what the first one that was most obvious to me was like, what functional role are these CAF EVs providing to PDAC? We talk, these cancer-associated fibroblasts are generally known to support, the epithelial cells that are in their microenvironment. And so we want to know if they can do that through the use, through EVs. And, then building on that, what molecular factors might be involved. So just to address this functionality of CAF EVs, this is just a basic schematic of how I'll be doing my functional assays, which will be to provide EVs from CAFs to serum-starved PDAC cells. So we starve these cells. I know this is pretty common for a lot of EV functional assays, but I think starving, pre-starving PDAC or these epithelial cells for 24 hours is especially relevant in the context of PDAC biology, because as I said, when you have this transition to this really activated cancer-associated extracellular environment, you get the stiffening and it becomes a very dense. There's a lot of interstitial pressure, which ends up collapsing blood cells or blood vessels. And so those environments are often very nutrient poor and often hypoxic. So they're very stressful for these cells, especially at early stages. And so, we starve these for the purpose of, recapitulating that serum stress, that nutrient deprivation stress that we potentially CAFs could rescue through the use of EVs. So just to, again, show you my methodology, the way that we will be quantifying survival and cell viability will be to take the ratio of living cells to a death signal. All of our PDAC cells that I use for these assays are ubiquitously expressing red fluorescent protein. So living cells will glow red, and then to mark death, we will use cytox green, which is a DNA stain that is membrane impermeable, so it's only going to stain dead or dying cells. And then I'm going to take the ratio of living to the death. And for most of my experiments, I will have my negative control being just vehicle treated, so PDAC only in starvation media. And then to begin, my positive control will be with conditioned media. So as you can see here, we have a pretty stark difference, as we would expect, that conditioned media does provide this rescue effect. And when we see if EVs can produce the same effect, we actually do, and that's really encouraging. And then I also wanted to see if the supernatant had any abilities to rescue these starving cells, and they do. And actually, they do have a degree of efficacy here, which appears to be able to supplement the EV fraction's efficacy to provide a more similar effect to the conditioned media, which I thought was pretty interesting. So that was a cool thing to kind of start off and see differences or effectiveness of our EVs to save these cells. But very early on, I just went back to look at what look at these individual channels to see if I can learn anything. And what I ended up seeing that really stood out to me, hopefully to you too, is that these cells are not so much becoming more proliferative as a result of EV treatment, but they are they're experiencing less death. And so to see, potentially that's EVs are helping to shield them from some type of death induction, I looked for the induction of apoptosis as cleaved caspase 3 as a readout for that. And so you can see that, yeah, in both the conditioned media and in the EV fractions, that, we lose this induction of apoptosis. So, potentially that's one way, one mechanism that EVs are shielding these serum-starved cancer cells from dying. But of course, that is the end of a mechanism, and we really want to know what, is further upstream and what unique protein factors could be, involved in this process. So that's going to bring me to our protein of interest that, star of the show. I have mentioned before, Netrin G1. So we initially found this protein's expression in the pancreas through a gene expression screen, comparing our normal fibroblasts to our cancer-associated fibroblasts. When we found this, we were really excited because it's a GPI-anchored extracellular membrane-bound protein, but it's involved in the stabilization of glutamatergic synapses. Now, it's pretty much only been described in neural tissue, and its expression in the pancreas is pretty novel. So I was like really, really exciting for us to pick up on. We see its expression also correlated with the onset of PDAC. Sorry, this is what it looks like. It's very similar to a laminin protein. There are a bunch of different netrins. However, Netrin G1 is this unique isoform, which I think is pretty special, especially in this context for reasons that I will continue to expand on in the next couple of slides. But before I do that, we can also see it, also this expression correlation with CAFs at the protein level. So you see it upregulated in CAFs compared to our tumor adjacent, which are more inactive fibroblasts. And then we're using U87 glioblastoma cells here as a positive control for its presence in the brain, being most common. Also, most exciting to me, obviously, as an EV researcher, is that we do see its expression in EV fraction. So that was really exciting. And, it continues to see this really cool data, matched or in vitro translating to the clinical level, because here we see that its expression specifically in the stromal compartments of the pancreas is virtually absent in healthy individuals as compared to the very strong expression and correlation with its expression in the tumor microenvironment in a tumor, like in the tumor region and even slightly in the tumor adjacent regions as well. So now this stromal Netrin G1 expression also has really interesting implications for informing prognosis. As you see here, using TMA data that we had from our institution, we see that Netrin G1 expression in fibroblasts correlate with a worse disease outcome in patients too. So there's a lot of evidence here that this is a really important protein in the context of the pancreatic cancer tumor pancreatic microenvironment. But what's even more exciting to me, I think, in the context of extracellular vesicles, is that when we look at the pancreas sections from spontaneous PDAC mouse models, specifically the KC mouse, which will develop PDAC tumors at around 40 to 50 weeks, we can see Netrin G1 expression in the pancreas beginning as early as 16 weeks in these KC mice, where we don't see it at all in the healthy wild-type mouse. Now, going back to the schematic, we know that these changes that are occurring, progressing towards tumor development are already happening at these early stages. So if we can see this Netrin G1 upregulation happening in these pan-in stages, much earlier compared to tumor development, detecting Netrin G1 in EVs, as a liquid biopsy could potentially become really significant when we think about monitoring the risk at-risk individuals for, pathological changes that are occurring in their body long before disease presents itself. And so I think, Netrin G1 significance is not simply to serve as a biomarker, because adding another piece to this puzzle, we see the expression of its sole binding partner, Netrin G1 ligand, or NGL1, also increased with PDAC induction. However, this time it's happening from the epithelial side of the relationship, so on the actual cancer cells themselves. So in vitro, we show that its expression is virtually absent in benign human pancreatic ductal epithelium, this HPNE condition, but that NGL1 expression is significantly increased in bona fide PDAC cell lines, in addition to pancreatic epithelium transformed with mutant KRAS, G12D specifically, mutant KRAS, which is the most common driver mutation in PDAC. And so the thinking here goes is that Netrin G1 is a membrane-bound protein on the extracellular side. It's coming out of CAFs. It's being used to it's classically described as stabilizing synapses, and it interacts with NGL1 on PDAC cells. So in the context of this cellular interaction, in this case, we think about EVs, we can think about these two factors as kind of being presynaptic and postsynaptic, which I think becomes really interesting, when we... So that becomes really interesting when we looking at these knockout conditions, where we're going to take, we're going to start knocking out either, one side or the other of this axis. So on our CAF side, we're going to knock out Netrin G1, and then on our our epithelial side, our cancer cell side, we're going to knock out NGL, and we're going to do these functional assays to see if EVs are still able to provide survival benefits. So first up, we're going to take EVs from Netrin G1 knockout cancer-associated fibroblasts and compare them to the EVs from our control CAFs. And here we see pretty cool that compared to EVs from our control CAFs, EVs from our Netrin G1 knockouts do not produce the same rescue effect. Importantly, we see that these cells secrete a pretty comparable amount of EVs. However, by ablating Netrin G1 in CAFs, you do end up getting differences in the distribution of EV subpopulations, which is definitely interesting and something that I would love to follow up on in the future. But now we're going to look at the flip side to that observation. When we knock out NGL1 on our PDAC cells and treat them with EVs from our control CAFs, we also see a significant loss in the pro-survival effects conferred by these CAF EVs. The same EVs are not able to rescue these knockout NGL1 knockout PANC1 cells compared to ones that do have that. So, taken together, these results become pretty exciting evidence, I think, that the engagement of Netrin G1 and NGL1, between EVs and PDAC cells is really important for the survival of this tumor, or these cancer cells. Now, like I said, I mentioned earlier that this Netrin G1 and NGL1 interaction has been described as stabilizing synapses. So we kind of wanted to take that one step further and think about this interaction as potentially being able to influence the engagement and uptake of EVs once they hit their recipient cell, in this case, PDAC cells. I think this would be really important to understand, because if the expression of these factors we understand to be upregulated with the development of PDAC, this axis could potentially exist as some form of mechanism by which PDAC selectively takes up EVs in other exogenous materials secreted by the stroma. So what I'm going to do is I'm going to take EVs from CAFs that ubiquitously express GFP, and I'm going to put them on our red PDAC cells. And we're going to observe differences in how these clusters of EVs are engaging the cells based on, the postsynaptic expression of NGL1. So here we see that not only, there are a lot more clusters of EVs in our control cells, but it also appears that they're, nice, evenly dispersed throughout the cell. And in contrast, we see that the EV clusters on our knockout cells are localized more to kind of like the outside. And, these 3D videos, I hope are able to come through clearly enough. But, we definitely see this yellow, a lot more yellow in our wild type cells, which would be like green material being like actively, packaged up and potentially, being processed through the cell, as opposed to in the knockouts, which we do not see that nearly as much. And so building on this observation, I wanted to see, if we could really, be more proactive about manipulating the interaction between NG1 on EVs and NGL1 on PDAC cells. And so the way that I am beginning to do this is by incubating CAF EVs with Netrin G1, recombinant Netrin G1 ligand protein. So the logic being here that this recombinant NGL1 will bind with Netrin G1 on EVs and effectively block them from making functional engagements with NGL1 on the PDAC cells that they are later administered to. And so very encouragingly, when we did this, we see that when we incubate these EVs with recombinant NGL1, we do partially lose some of the pro-survival efficacy of our EVs that we see in just like the control condition, which is really exciting. And now I plan, this is kind of where COVID, the lockdown kind of left me, but I definitely plan to follow up on this observation by using EVs from a CAF cell line that we generated with a mutation in Netrin G1's binding site, so as to prevent it from making functional engagements with NGL1, as well as using a small peptide that targets NG1's binding site to do something similar to what I tried to do with this recombinant protein. But so far, that's where my project stands on the functional significance of EVs front. So I'll just kind of share some of the strong takeaways that I have, being that cancer-associated fibroblasts produce these EVs that are providing these pro-survival effects to nutrient-deprived PDAC cells. We know that NG1 and CAFs and NGL1 on PDAC cells are needed for CAFs to be able to confer these pro-survival advantages through their EVs to PDAC cells, and that disrupting this interaction could potentially impair EV uptake and efficacy, which I think in the most optimistic sense, this is definitely, I think, out of the scope of my PhD project, but thinking about the implications of these observations, I think they could definitely maybe serve for like the groundwork for approaching therapeutic interventions, either by blocking the uptake of NG1 positive EVs or by using Netrin G1 on synthetic liposomes as a homing device for potentially like drug delivery, or to find where Netrin G1 ligand might be in the body. And so to the point of really, so leaving the functional stuff there and then really trying to get towards what I think a lot of people in the field are trying to do, which is to really understand EVs on an individual basis, because we do understand that there's such a large amount of heterogeneity that exists in the EV populations that we're isolating, even though we are enriching for specific size range, we still understand them to be very biochemically distinct. There are some really wonderful studies aiming to do the single EV analysis, which I think would be really monumental in when we think about pairing the data that we get from there, especially in like liquid biopsies, to information that we understand for specific organ biology, and the applications for like technology like that, and screenings and all that stuff is really, like, for me as a student, really mind-blowing. So my investigation is a lot more humble than that, but I'm going to take you through how I've been personally trying to dissect the heterogeneity of our CAF EVs. And to do that, I think it'd probably be best if I quickly come back to our cell model, just so I can introduce one other relevant protein of interest. This protein being an integrin pair that you see labeled in red. Our lab has studied this alpha-5 beta-1 pretty extensively for its role in activation states of pancreatic, in the different activation states of our pancreatic fibroblasts. As you'll be familiar with by now, in our inactive state, we see Netrin G1 pretty absent, where we do see it being expressed in bona fide CAFs. But we also see differences in this integrin's localization to be dependent on activation status. So in an inactive state, this integrin, when a CAF is in an inactive state, this integrin is found at the plasma membrane. But during this fibroblast activation, it's shuttled internally via endosome trafficking pathways, which land them in the multivesicular body, and unsurprisingly, as I'm sure many of us would say, being packaged into exosomes. So we have these two factors, and to begin investigating the EV heterogeneity, I wanted to just see, using electron microscopy, where these proteins, what types of EVs these proteins are finding themselves in. So, like I said, with this integrin, we know that it's pretty common for integrins to find their ways into exosomes. So, unsurprisingly, we see this integrin pair co-localize with CD81. This is using immunogold labeling, different sizes. So we have a larger particle here compared to smaller ones for the different proteins. But looking at Netrin G1 next, we see something really cool. And this was probably one of the most exciting things that came out of this, just this part of the study, was that the EVs that we see co-localizing to Netrin, or Netrin co-localizing or localizing to, are very distinct from that of an exosome. They're much smaller. They look single membraned as opposed to double. And when we continue to do dual stains of Netrin and this integrin, we see them almost never localizing to the same EV. This is a quantum dot staining, so it's a little less electron dense. But importantly, these are representative images, and we never see them, localizing to the same EVs. And further, we never really see Netrin co-localizing to an exosome structure at all, as you would see, in this bottom right. So, these are pretty interesting hints that this Netrin is being secreted differently. There's different biogenesis and potentially different cargo implications in these Netrin G1 positive EVs as compared to your canonical exosome, or where this integrin pair is going. But these are still just representative images, and we really wanted to know, is Netrin G1 indeed enriched in these tiny EVs, which some, depending on what study you might call them exomeres, distinct nanoparticles. I wanted to kind of see, on a prep-wide basis, how we could, split these subpopulations and see if Netrin is truly enriched in them. And so to do that, I found inspiration in this paper from last year, which I really liked, in which researchers use this extended ultracentrifugation step. It just goes overnight. So you're kind of further separating EVs from that original SEV pellet. And so I did that with my CAF EVs, and just kind of initially looking at the NTA data, we do see this enrichment kind of left skewing in our distinct nanoparticle pellet. You have increased size ranges, and the very small, between 40 and 80 sizes compared to the SEV pellet. But what was really, the most encouraging result is that we do see, in fact, Netrin G1's enrichment in this distinct nanoparticle population. And again, I was also happy to see that my seeing hexokinase being enriched in the DNP versus our more classical exosome markers being enriched in the SCV population, very consistent with what that paper showed. I was pretty happy to have all this, these consistent results show. And just to kind of further emphasize this enrichment in what we would call, exomeres potentially, I took the ratio, the internal ratio of Netrin G1 to CD81 within each fraction, just to kind of, show that we are enriching Netrin G1 in these smaller cell EV populations. So, this is also where the kind of shutdown, coronavirus shutdown hit me, and I'm really excited to get back to this specifically, so I can really test the functional effects of each of these fractions in my survival assays to, really understand the importance of Netrin G1 positive EVs in this system. But something that I thought that we could talk, I really wanted to kind of emphasize, because it's not, these Netrin G1 positive EVs and finding Netrin G1's enrichment in them is not the only interesting thing, I think, when I'm thinking about, what makes these EVs special. And, to kind of emphasize that, I want to come back to thinking about, the extracellular environment that I described earlier in my talk, and, how it can have a profound impact on how we, think about the fate of secreted EVs in pancreatic cancer, or really in any in vivo context. And so initially, I was really interested in seeing how the extracellular matrix was produced. The extracellular matrix that's being built by these CAFs is affecting the EVs that are being secreted, into that extracellular environment. So to visualize this, I used immunofluorescence staining technique that we've done in our lab a bunch, to use, in these 3D cultures to see what's on the outside extracellular compartments and what's intracellular. So what I'm going to be showing you, you'll be seeing fibronectin labeled blue, which is like the extracellular matrix. And then you'll also be seeing these green clusters of either CD81 positive or CD63 positive EVs that are trapped in the extracellular matrix. And so I can show you, especially if you look at, these merged photos, you can see, that these different populations, either, CD81 positive or CD63 positive have very different localization patterns. And, what this really means to me as an EV scientist, and, something that I hope to be able to provide as food for thought, to everyone here, and just to be able to discuss this with you, is that, there are, when we think about really like the in vivo context, there are these almost filters to what populations of EVs are being collected when we take liquid biopsies. So, for example, just in the context of my study, this integrin alpha-5 beta-1, its main binding protein is, binding ligand is fibronectin. So that's the main component of this ECM that you're seeing stained here. So could we be missing a sizable population of EVs that are positive for this integrin because they're being trapped in an extracellular environment? Also thinking about the size of these particles in this environment, which I talked about being very dense and very hard to travel through, because smaller EVs, like the ones that are Netrin G1 positive, might be able to slip through into the bloodstream more effectively than larger structures. Then again, because of Netrin G1 and NGL1's interaction, could there be a selectivity taken up by this PDAC and make, and in that sense, these EVs are being selectively taken up by PDAC in a paracrine manner and not making it into circulation at all. So I just think, there's a lot to think about. That's, kind of where my brain's been exploding. And I really love watching and being a part of these discussions to really kind of help me get through, make sense of that kind of stuff. These are just a few things that I'm trying to wrap up for my degree in the next year. I'm probably a year away. I'd really, I'd be happy to talk to you guys about any of these points. But I will just highlight one exciting avenue that I have some proof concept images to look for Netrin G1 positive EVs in human blood, which I'll be doing as part of a clinical trial we have coming up at my institution, where I'll be analyzing EVs from blood and from patients before and after radiation therapy, which is aimed to normalize the tumor microenvironment from an active to a non-active state. So being able to not only look for them in these liquid biopsies, but potentially do the fractionation of subpopulations and be able to look for them in that way. So yeah, these are just proof of concept. Also, be happy to take any tips on how to improve looking at EM images of things come out of the blood, because as you can see, especially here in the human, there's a lot of like background that messes up the contrast and all that. So with that, I just want to give you some take-home points from my talk, and kind of put together show you this working model that I've been putting together. Specifically, CAFs, they're secreting these EVs with these PDAC associated factors, Netrin G1 and our integrin alpha-5 beta-1. We see these proteins are found in separate types of EVs, which likely implies different biogenesis and potentially different functional significance, as well as cargo. EVs from CAFs are able to protect PDAC cells from nutrient deprivation induced apoptosis, potentially. And when we disrupt this NG1 NGL1 interaction, these EVs, the efficacy of our EVs to support from CAFs to support PDAC is greatly diminished. And again, I think I really can't stress enough how much I've been thinking about really just how we can start thinking about cell of origin, and just factors that influence EV cell fate, whether what stays local, what makes it to a systemic, and everything that can kind of influence those two aspects. This is my lab. They are fantastic. That's our lab. Twitter, no such thing as too much self-promotion. They're a really great group. I've been in a few different labs, since undergrad, and this is by far the most supportive one I've ever been in. I really want to pick out Ralph from the crowd. You've seen a lot of his data, especially in the Netrin background data that I was showing. He has a paper out with his wife, Deborah, who is right below him. They're co-authors on this paper that is really excellent, and we're all really excited about it coming out. And yeah, so with that, I'd be more than happy to take questions, just talk about this investigation, what things I'm learning, anything. Thank you very much.
Thank you so much, Kris, for an amazing talk, and congratulations on wrapping up your PhD hopefully soon. That was amazing to hear about your research and your lab. There are a series of questions for you. We're going to start with Alisa. Alisa, do you want to go ahead?
Question about whether the EV supernatant really had removed all the EVs from it, because as you may know, it takes a long time to really centrifuge all the EVs down. And then I had another question that I didn't post about, it looked like the Netrin G1 was possibly inside of your small extracellular vesicles from the EM. Have you looked at the topology of where the Netrin G1 is?
Well, thanks for your questions. So the supernatant, I am pretty, I feel pretty confident that I've eliminated, as much as possible the EVs from there. I've done, I probably should have pointed it out, but the conditioning media, I've made sure that, that's free of EVs, but also, yeah, I mean, the supernatant, especially earlier on in my studies, I put a lot of emphasis on making sure the purity of that fraction. In terms of whether Netrin G1 is inside or outside, I guess my best answer is that because doing the EM, we're not permeabilizing those EVs in any way, that the immunogold particles are sticking to the outside of those cells. And so I would consider them extracellular in that way. But if you have any input, I'd be happy to think about that.
No, it was funny to me because it looked like they might be inside, but of course it's really impossible to totally say from EM, and your functional studies certainly suggest that it's on the outside and mediating interaction. So I was just curious. Great talk,
we do know, thank you. I will say that we do know that, Netrin G1 is only extracellular, so it's not transmembrane and it's not found on the cytosolic side of the plasma membrane. So by that, we feel pretty confident that we're just seeing it on the outside of these EVs as well.
Sure.
Thanks. Like, the conversation has been so exciting. My son decided to join us for the wrap up. Flip, do you want to go next with your question?
So my questions have progressed. Kris, it was a very warm and wonderful presentation.
Thank you.
That I really enjoyed. Now, the recombinant protein, you could make a lot of that, I think. And then if you had a mouse model, you could see if that interfered with the metastases or something like that. And then the other thing to do with it is to make an antibody and do an affinity chromatography on the EV to at least sort out the positives and maybe subpopulations that way. It's a very powerful technique. That would be an affinity column.
Yeah.
Batch is no good. A column is like a whole series of batches. So I don't know if you could use the protein on the column. That would be for the target cells. But I think the antibody would be for the EVs. Do I have that right?
Yeah, no, absolutely. And that's a great point. Ralph's study that I mentioned, he was doing a lot of in vivo work, and we actually he had this one really fantastic result that we did just that. We injected a anti-Netrin G1 antibody into this mouse, tumor bearing mouse, and we saw a reduction in tumor size, and so that was really exciting. So I'm definitely going to try to recapitulate that in the context of my study. But to your point about the affinity chromatography, just to understand that binding, I definitely would love to. I'm definitely not a biochemist by training, but I would love to get in touch with researchers that would be able to really help elucidate the kinetics there and the binding affinities. Because I think, especially like I said, the kind of long-term aspirations of these types of findings would be to produce something that we could inject into in vivo context to neutralize either Netrin G1 EVs or its recipient cells from taking them.
Well, you already have the antibody, so the affinity columns is not a really complicated biochemistry, and then you can elute the EVs because they tend to be very resistant to pH. You can use acid or base to elute them, or maybe both, and see what you get.
Awesome. Yeah, thank you so much for that.
Yeah, if you elute with acid, then you have neutralizing base in the tube they're eluting them into. If you use base, you have neutralizing acid in the tube you isolate them in. So I'd be glad to talk to you further about that anytime you'd like.
Yeah, absolutely. I'll definitely reach out. Thank you so much.
Okay, next question, Kalyani. Are you ready to ask your question?
Yes. Hi, Kris. It was a great talk. So I have a couple of questions here. The pancreas has neurotransmitters. So, and you were talking about the presynaptic and postsynapses. So, do you think that the neurotransmitters in the pancreas, or have you looked upon the effect of neurotransmitters on the EV-associated expression of Netrin G1? Because what you have found in the Netrin G1 expression is quite novel in the pancreas. So have you found any factors that may affect the expression of Netrin G1?
Yeah, no, that's a brilliant question. So we do have kind of part of the larger context of our lab study. Like I talked about that integrin pair. We see that when we, mess up certain, these cells' interactions with its extracellular environment, the expression of Netrin changes. Its activation state we can change with like TGF beta, either by blocking that or providing it as a ligand. And yeah, we see Netrin G1 changes a lot in those contexts. The pancreas is a really interesting organ, especially in the context, I think, of when you talk about neural tissue, because there is, especially during disease progression, this perineural invasion, and it's really incredible. I mean, you can even, like metastasis, occurs like kind of along nerves, which is wild. And so, yeah, of course, that's the first thing, one of the first things we thought about when we were seeing these neural proteins in the pancreas. There must be some type of interaction there with, the nerves themselves, or, some type of synapses. I mean, we see glutamate and glutamine synthesis and conversions to be really, like there's definitely like something significant there. Ralph, that's also in Ralph's study, looking at, when we knock out Netrin in CAFs, that we see differences in glutamine synthesis and the enzymes that, have to do with that.
Sorry, I just another question.
No, go ahead.
So this is in continuation with what Philip suggested, that about metastasis. So you looked into the expression of, you looked into the effect of integrins, and as you may know that there are an array of integrins that have been found in pancreatic cancer. So one integrin that you specifically looked upon was alpha-5 beta-1. So in continuation with this, I have a question that pancreatic cancer, if I'm not wrong, has preferential metastasis in the liver. So, as pointed out by Hoshino, he says that according to them, the alpha-V beta-5 integrin plays a crucial role in the organotropic metastasis in the liver. So, do you think that looking at the expression of that integrin in association with Netrin G1 would give you a better idea about liver metastasis, because you have already done such an extensive work in Netrin G1 and using the integrin alpha-5 beta-1? So, do you think that alpha-V beta-5 can be another potential candidate to look upon?
Yeah, I'm like watching my PI laugh, but it's because, so we do actually have a whole system there, because, so one way that we've been able to, I'm sure many of you know that primary cells are very tough to maintain their phenotype and their integrity. So that's very much so the case for normal fibroblasts. So a way that we've been able to continue to study these activation states has been by creating a genetic mutant of our CAFs, specifically in the beta-5 alpha component of the alpha-V beta-5. And so we see, and this Janos, who is also on this talk, he has a paper in eLife that gets into all of this, the interaction, the relationship that exists between alpha-V beta-5, alpha-5 beta-1, and promoting the different activation states in the fibroblast and the tumor microenvironment, and where we see this also in colorectal cancer, I believe we see similar trends. Yeah. Sorry, to your point in the liver, I'm not specifically sure about the context in the liver, but I definitely would imagine there was a similar relationship.
Thanks, Kris and Cleone. Next question is from your colleague, I think, Janice.
Hi. In very impressive job. I should not say this because you're my lab mate, but yeah, you did have a very good job. So Have you checked the presence of the receptor of Netrin G1, so Netrin G1 ligand in the EVs that contain either alpha 5 beta 1 or the other type of EVs that contains Netrin G1, if it's any colocalization of these two in particular subsets of EVs?
Yeah. No, I haven't looked specifically for done co-localizing, co-staining of the ligand and the integrins. I think there's definitely, like, I would love to be able to do, I mean, I still can, do like pull down assays, pulling down different, you know, either the integrin or Netrin, and seeing, you know, if we get similar co-localization there. I really, you know, every time I watch a single EV analysis study, all I can think about is, oh my gosh, like, I wish, you know, like, just to be able to, like, understand what, you know, we can tell and pick apart from those. But, you know, definitely in the pipe. Thanks.
Okay. Thank you.
You can do with flow cytometry in bulk.
Yeah.
And to try and as a population that stains with both.
Yeah, I mean, so forgive me if this is naive. I was under the assumption that flow cytometry for EVs was not as wasn't able to tell you too much about size populations, or you know, like there's they're too small to
but we're pretty primitive, and
yeah,
with EVs stuck to particles, you can then stain them with antibodies.
Sure.
And determine whether you've got a co-staining. You first select out the particles that only have one or a few EVs on them.
Right. Okay.
And then look for double staining. It's very sensitive.
Yeah, actually, no, I mean, that's definitely something I could start looking into, because I would love to be able to, flow cytometry, I think, is, you know, like a great application.
Absolutely. Yeah.
Thank you.
Okay, and last but not least, Dolores have a couple of questions for you.
Yeah, I have a couple of questions. First of all, I really enjoyed the presentation. It's great, and it's nice to see, you know, people who haven't been working on EVs forever joining the field with their innovative approaches, with so many interesting models and stuff like that. So I really enjoyed it. I have a question that is unrelated to your presentation, which is, what do you think? So if I understand correctly, your NG1 is not expressed in the pancreatic cancer cells at all. It's only coming from the...
Yeah, we see it in the stromal compartment, specifically cancer-associated fibroblasts.
So it cannot be the same protein, but do you have any... what do you think the cancer cell-derived EVs can do to the alignment of the extracellular matrix? Do you have any data about that?
No, so I think that that's absolutely like a brilliant question, because like I was saying, you know, when we think about this whole system, there are signals being sent from every cell to, you know, potentially every other cell that surrounds them, on top of them just being stuck in the matrix, you know, and you know, potentially, you know, being available for you know, chemotaxis or whatever, you know, however the context might work. But I would expect that, you know, there have been plenty of studies that show that, yeah, cancer cells can, you know, activate quiescent fibroblasts and initiate this tumor, you know, associated phenotype, you know, and it's definitely, like, you know, depending on the disease, it's a very chicken or the egg question, because it's so hard. I mean, especially in the pancreas, it's so hard to know, you know, did the environment start changing, or was there like a, you know, like a spontaneous, you know, driving mutation? You know, tumors in the pancreas are generally not as heterogeneous as many other tumors in other parts of the body. It's pretty much just, you know, P53 and KRAS drive these cancers. But to that point, you know, the microenvironment and the heterogeneity that exists in the microenvironment has such a big influence there. So again, it's definitely like a back and forth.
Yeah, no, it would be interesting to use your system to test the role of the pancreatic derived EVs on the matrix. I really like that idea. The other quick question, I'm sorry we're late, but I wanted to mention one thing. So I think at the end of your presentation, you almost alluded to the possibility that the integrin alpha 5 might be in the exomer together with NG1, although I don't know if you are hypothesizing that they work together or not. And I wanted to say that we have done a lot of proteomic analysis of large oncosomes and exosomes, and all of the integrins are in exosomes except for A5, alpha 5, which is in large oncosomes predominantly, at least in prostate cancer derived EVs. So, I mean, do you think it's in exomers in your system?
No, I'm sorry. Maybe I misspoke, but I specifically see them in different EVs. So I see Netrin in the, you know, quote unquote exomers, and the alpha 5 beta 1 integrin almost, you know, exclusively in the exosomes.
Okay.
Yeah, and so our lab also, you know, does a lot of work with this integrin, and when I talked about the localization being different, we think that, you know, when it's activated and it's being shuttled inside the cell, of course, it makes sense it ends up in exosomes. But we also have this, you know, idea that maybe when it's on the outside and at the plasma membrane, it could potentially be being pinched off in microvesicles in that context. So, you know, again, it definitely goes to, you know, the onus is on us to understand the cellular context so that we can understand where, you know, these proteins are in EV, you know, the types of EVs that they are.
Okay, that's great. I think we can, Elam, there are no more questions, right?
No, that's it.
So I think we can close this this session. I just want to thank Kris again for a brilliant presentation, and mention that next week we will have Luca Musante from Uta Urdbrugger Lab in Virginia, and he's going to talk about urinary extracellular vesicle analysis and pre-analytical variability.